First off, let me thank Leo and Al for inviting me to be a part of this symposium. I'm really excited to hear all the different talks during our time together. So I'm going to tell you a little bit about genomics and hybridization in vermivora warblers. My name is David Toews. I'm an assistant professor in the biology department at Pennsylvania State University. But before I begin, I want to suggest you don't necessarily want to just be listening to me. I think you'd rather be listening to these superstars. Marcela Baez looking at microbiome variation across wood warblers, Steph Zarmak looking at genomic divergence in yellow-rumped warblers, and Lanny Feng quantifying bioacoustic and genomic variation in Nashville warblers. So if you get a chance, I would strongly recommend taking a look at their talks as well. So like I said, I'm going to talk about vermivora warblers. So this is a small 3-species genus within the New World wood warblers, consisting of Bachman's warbler, golden-winged warbler, and blue-winged warblers. And nothing is ever simple in the world of vermivora warblers. So Bachman's warbler is one of the few continental bird species in North America presumed to have gone extinct over the last century. Golden-winged and blue-winged warblers hybridize extensively where they co-occur in a hybrid zone unlike others we've seen before. So today I'm going to first talk about golden-wing hybridization between golden-wing and blue-wing warblers. And to begin, I'm going to introduce a bit of a ham-handed metaphor. In this case, I'm showing the two sides of my reproductive isolation coin, where isolating barriers on one side can impede gene flow introduced via hybridization on the other side of this coin. And so now, obviously, on a symposium on reproductive isolation, we would rightly be interested and focused on isolating barriers. Over at least in golden-wing and blue-wing warblers, we know much less about these isolating barriers than we do the dynamics of hybridization. In hybridize, they do. So here I'm showing you a distribution of golden-wing warblers, which occur at higher latitudes and elevations throughout the Appalachians, and blue-wing warblers shown further to the south and at lower elevations. Where these two species come into contact, you can see a range of hybrid phenotypes, which I'm showing on the illustration, showing with these illustrations. Pretty much anywhere they co-occur, and it's patchy and generously described as a mosaic hybrid zone. Folks have been studying these birds for years, many years, in fact. So I'm showing several papers from the early 1900s by Nichols and Faxon, who were both interested in describing the different possible phenotypic outcomes of hybridization. More recently, Frank Gill compared the phenotypic composition at different geographic localities and at different time points. But by necessity, as I've emphasized, these past studies have really just focused on phenotypes. We really had no objective marker to diagnose the parental types or hybrids until our whole genome analysis in 2016. So here I'm showing you a Manhattan plot of differentiation across the genomes of blue-winged and golden-winged warblers. And the exciting part about the study was not only were there only 6 regions that were highly differentiated, but most of these regions were near genes that had some strong association with plumage pigmentation phenotypes. But what I want to ask today is, what else can we learn about hybridization now that we're armed with these genetic markers that can distinguish the species? So one thing we can do is estimate hybrid composition in the hybrid zone using triangle plots. So here I'm showing you an example of some simulations that I did for another hybrid zone. The idea here is that you have a hybrid index along the x-axis ranging from species A to species B. On the y-axis, you have interspecific heterozygosity. So in this case, the maximum heterozygosity would be 1, which is where the genome is highly heterozygous, like what we would expect in an F1 hybrid. And then depending on which direction those hybrids backcross will determine which side along the triangle that they'll fall. And if there's significant hybridization within hybrids, they'll fill out the middle of this triangle. And this is a really wonderful way to illustrate the complex composition of different hybrid zones. And in a paper a couple years ago by Pulido Santa Cruz et al., they did some extensive simulations about how different types of selection or reproductive incompatibilities would generate different types of these triangle distributions. And the 5-second moral of this story is that with more selection against hybrids and more incompatibilities as we move along the right, there are fewer individuals that fall in the center of this triangle. So how do these look in vermivora, and can we learn anything about the dynamics of hybridization? So a couple points before I get in here. So here we're looking at 6 SNPs in the divergent regions of the genome. So this hybrid index that I'm using along the x-axis in vermivora isn't really an unbiased summary of the genome-wide patterns. However, these analyses require us to work with nearly fixed differences. Also, because there are only 6 SNPs, there are a lot of overlapping points. So I'm going to show you these triangle plots where these large points, the opacity of those points actually represent the number of individuals in that bin. And I'll also note here that Marcela Baez has a paper where we did something similar with triangle plots using genomic data and not relying on these 6 SNPs. So when we look at allopatric populations, in this case for blue-winged warblers, it's Missouri, and golden-winged warblers, it's Manitoba, we don't really see any evidence of hybridization. There isn't really opportunity, and most of our birds fall within the lower corners of the distribution. By contrast, here in the hybrid zone, in this case, these are birds from across New York State, where there's lots of overlap, we see a much different pattern. We see a much more filled out triangle with lots of evidence of hybrids. In fact, if we compare all those birds that have a high interspecific heterozygosity, so higher up in the triangle, evidence of hybridization, compared to birds in the region that falls closer to the parental types, so the bottom corners, we see that almost 40% of birds have some evidence of hybrid ancestry. By contrast, if we just looked at those birds that researchers in the field classified as one of the various hybrid phenotypes that I talked about earlier, only about 15% of them were classified as hybrids. So already our genetic data is giving a much more nuanced look at the extensive hybridization that occurs in the system. But how has hybridization influenced genomic patterns more generally? Well, when we first published our paper in 2016, we didn't have much to compare to, but we now have data from many other species pair, in this case in Cetophaga, the Cetophaga genus, where we don't have evidence of hybridization. So for example, here I'm showing you our nuclear Manhattan plot of differentiation in vermivora. Importantly, non-recombining mitochondrial DNA shows us that these species have had a fair bit of time, or had a fair bit of time in isolation, over 3% mitochondrial divergence, which would have required about 1 million or so years in isolation to accumulate. By contrast, Grace's and black-throated gray warblers, which are sister species, have overlapping ranges in the Southwest, no evidence of hybridization, have only about 1% mitochondrial divergence, but look at the difference in the patterns of differentiation in the nuclear genome. Average FST is much, much higher, many more peaks of divergence, and this is consistent with strong reproductive isolation. Point here being that hybridization and introgression has had a pervasive influence on the genomes of vermivora warblers. So going back to my metaphor, I've really focused on the effects of hybridization in the system because it's proved easier to quantify and understand. However, there are some researchers that have been more focused on different isolating barriers, more specifically the role of assortative mating and social pairs. Now, as I'm showing here, there's been a fair bit of back and forth in literature about these studies, and I encourage anyone that's interested to take a look at this dialogue. However, there's several issues with looking at plumage-based social pairing, not the least of which is high extra pair paternity in this system. But I'm mostly going to leave it there in the academic literature, but it does bring up a broader point about perspectives and interpretation of the data in this system. And in the years since publishing the genomic comparison between golden-winged and blue-winged warblers, it's been interesting to see how different people have interpreted these findings in different ways. So going back to my stupid coin, some have suggested, wow, you've found the speciation genes that keep these two species distinct. Whereas other people have focused more on the genome-wide patterns of homogeneity, suggesting that these things are so similar that they're probably not even distinct species. And I don't really have an answer here, but I think this is still the big question that is outstanding. What is the extent of reproductive isolation in the system, and how have these differences, which are real, been maintained in the face of gene flow And I think it's going to require a combination of field studies of assortative mating, pedigrees, genotyping to really understand how these differences are or are not being maintained in the system. And so for the last minute or so, I'm going to switch gears and talk very briefly about Bachman's warbler. This is an enigmatic species that was last observed in 1958. And we were actually able to extract DNA from a skin dating back to 1890. We're now talking super low coverage and an N of only one individual, but we're working on getting more data. But Andrew Wood in my lab has been interested in testing whether there is any evidence from this limited sample of introgression between Bachman's and golden wing and blue wing warblers. And there's a lot here, but what I'm showing is a principal components analysis of the various divergent regions between golden wing and blue wing warblers. And the game is really to follow the red dot, which is the Bachman's warbler. So 2 fun points. Along PC1 in several regions, Bachman's falls intermediate between the 2 clusters with other contemporary hybrids. And second, BCO2, Bachman's clusters strongly with blue wing warblers. BCO2 is one of these cases. It's involved in carotenoid deposition and processing, which shows strong introgression among several species of Psittophaga warblers. So this could be a signal of introgression of BCO2 among different vermivora as well. But we note this is all very preliminary, and we're looking to generate more ancient DNA before making any more strong conclusions. And so with that, I'll summarize. The genetic data shows that hybridization is extensive between golden wing and blue wing warblers. Preliminary analysis of ancient DNA shows that Bachman's might have gotten in on this game too, also showing some possible instances of introgression. And taken together, if you believe all this, vermivora warblers don't seem to have been very good at evolving reproductive isolation, even given plenty of time. So with that, I'll put up my acknowledgements and thank you very much for listening to this presentation.